Okay, so first gene mutation we're going to look at is substitution. So you can see from this diagram, I'm trying to explain in kind of the sequence of events in relation to how do we get our mutation, our overall mutation. So remember what I said earlier, so when we talk about substitution, we're talking about the change in this nucleotide sequence, okay? But we also have to consider the effect on the amino acid. And when we talk about amino acids, remember amino acids are the building blocks of proteins. So when there's an issue with the amino acids, there's obviously an issue. There's going to be a mutation or an alteration to the overall protein. So as we know, we have a sequence of nucleotides within our DNA molecule, okay? And what happens, there's a copy of that, which is therefore a messenger RNA, and our messenger RNA goes to the ribosome where it meets transfer RNA and it creates the protein, okay? So when we look here, so this is this particular sequence, so this is the triplet of bases, okay, that codes for this particular amino acid. So we're going to call it lysine, okay. So TTC is our 3 nucleotides. Messenger RNA, okay, is going to be synthesized, and that sequence is therefore going to be AAG. And remember, this is our codon. This is what's classed as a codon, okay. So the messenger RNA goes and travels to the ribosome, meets transfer RNA, which is carrying the amino acid, and therefore part of this protein is going to be this amino acid called lysine. So that is what we're expecting. However, what happens is, during our substitution mutation, instead of TTC, which is the correct sequence of nucleotides, we now have TCC. Okay, so this T has been substituted for a C, okay, which therefore gives us a different sequence for our messenger RNA. Okay, so no longer is it a T, it's a C. So remember, complementary base pairing. What's going to happen is we're not going to get what we would like, AAG codon, we're going to get AGG, because again, G guanine pairs with C cytosine. So this gives us a different codon. And because we've got a different codon, we therefore end up with a different amino acid. Okay, so we're going to end up with arginine. So this is what's classed as our missense. So the effect of a substitution mutation on the overall protein. So as it says here, missense, the altered codon, so this altered codon, because we no longer have what the original sequence should be, codes for an amino acid that still makes sense, but not the original sense. So the original sense is lysine, but we now have this altered sequence, i.e. this different amino acid, okay? And what we tend to say with missense is it really depends on the type of amino acid that has been synthesized instead, and whether it's what kind of effect it has on the individual in relation to that overall protein, okay? So sometimes it won't have an effect, sometimes it will, but it won't be fatal or it won't cause major health issues, and then other times it can be quite severe. So it just really depends on the role of that protein, okay? And then the second effect that a substitution mutation could have is what's classed as a nonsense mutation, okay? So again, this is what we expect, this is what we normally would have, but with the substitution mutation, so this time what's happened is we got a sequence of nucleotides, and instead of having TTC, which is a normal one, we now have ATC, okay? So this thymine nucleotide has been substituted for adenine. And again, because we've got a different sequence, we therefore get a different messenger RNA. Okay, we get a different sequence within our messenger RNA molecule. Okay, so again, complementary base pairing. So we have our adenine, so that therefore means that's going to be uracil. Okay, so remember, no thymine with RNA, it's uracil instead. But what's happened is, although we've created a codon for ourselves for a messenger RNA, what's happened is that sequence of nucleotides therefore gives us a stop codon. Okay, and remember, we need, so when we're creating our messenger RNA, we need a start codon and a stop codon. Okay, And that stop codon therefore tells the cell, okay, to stop, or the ribosome, to stop producing any more, okay, any other extension to that protein. So because we've got a stop codon, okay, we have a smaller sequence, and that smaller sequence is enough to provide this mutated version of the protein. So as it says here, a nonsense mutation, an amino acid codon is substituted with a stop codon, okay? So substitution mutations, this is how it works. This is our normal, what we would expect in relation to what kind of amino acid we're going to get, and i.e. what kind of protein. And then when we get a substitution mutation, we can either get a missense effect on the amino acid and protein, or nonsense. And the next one we'll look at is the third one, which is a splice site mutation.